All right, shall we start now? Or participants are still joining?
Yeah,
So,
we can wait. No problem, Archana.
okay, a couple more minutes and then we'll start.
Yes, sure. Once you start, that recording button will start blinking, right?
It is already in recording mode. I turned on the recording, so it's right now in recording mode.
From here, I can see it's not blinking. That's what I'm just asking.
No, it is live and recording. Both are. I
Okay.
turned it on. Yes. Yeah. All right, so let us start without any further delay. Very good morning, everyone. All the participants, all the faculty members, and our guest of honor for today's event. First of all, I welcome all of you on behalf of PP Savani University for the international webinar. PP Savani University and AIMST University became collaborative partners with a goal to promote academic and cultural exchange. In February 2020, students from PP Savani University visited AIMST University under Global Immersion Program this year. Okay. In order to further strengthen our academic ties, we are jointly organizing this webinar. The ongoing pandemic situation not only has resulted in loss of millions of lives, but also has badly affected the global economy. One of the primary causes for this situation is our lack of awareness towards biosafety and biosecurity and biorisk management. That is why it is highly relevant and important to make ourselves aware about biosafety, biosecurity, and biorisk management. Therefore, PP Savani University, in collaboration with our international partner AIMST University, is organizing an international webinar on an overview of biorisk management from One Health perspective. All right. So now I would like to welcome our expert speaker of the session, Dr. Subhash Bhore. I'm going to introduce a brief intro about him. Dr. Subhash is IFBA certified professional and senior associate professor in the Department of Biotechnology at AIMST University, Malaysia. Dr. Subhash completed his BSc and MSc in Botany at University of Pune, India. In June 2000, he had received fellowship to pursue his PhD in molecular genetics at National University of Malaysia. Following his PhD, he worked in various key positions in the area of biotechnology. Currently, he is working as a senior associate professor at AIMST University. He has published several peer-reviewed articles, books, guided a lot of graduate and undergraduate students, and he has received more than 18 awards and fellowship at several international levels. He is actively involved in teaching, research, and advising students, undergraduate as well as postgraduate. In 2017, he earned his professional certification in biorisk management from International Federation of Biosafety Association, Canada. As a certified professional, he is actively promoting biosafety and biosecurity And last year, for the outstanding work at AIMST University related to 2019 Biosafety and Biosecurity Month, he received an honorable mention in International American Biological Safety Association's annual conference held in November 2019 at Alabama, USA. So for today's event, we have global participants from various parts of India as well as from Malaysia and Indonesia. So participants, I have also mentioned this one before, that you will get a certificate of participation once you are going to fill the feedback form, which I'll share at the end of the event. I would also like to request all the participants to kindly keep their microphone mute throughout the session. If you have any question or query, please feel free to write in the chat box. All right. So right before we start, and I'm going to invite my guest of honor, I just want to make an important announcement for all the participants about this event. Okay, so our partner university, AIMST University, is observing October 2020 as a Biosafety and Biosecurity Month with the goal to raise biosafety and biosecurity awareness. So beside today's event, which you are all participating in, they are also having 9 other webinars and one e-poster competition, which is going to be held on 30th October at 10:30 a.m. Malaysian Standard Time and 8 a.m. Indian Standard Time. Okay. So I really would like to encourage all the participants and staff members to participate in that, because you will be having cutoff marks, and based on your grades you will get A, B, C, and all the participants who come in A grade, B grade, or C grade, they are going to receive gold, silver, and bronze medal accordingly, okay, respectively. So if A, gold, B, silver, C, bronze, along with the certificate. So this is really good opportunity for you to get a good learning experience as well as get an award. Now, before we start our expert talk, I would like to invite our guest of honor, Senior Professor and ex-Vice Chancellor, Dr. M. Ravichandran, Dean Faculty of Applied Sciences, AIMST University, Malaysia. Sir, please, over to
Yeah.
you.
So good morning, everyone. I think hope you all can hear me. And thank you, Dr. Archana, for a kind introduction. And on behalf of AIMST University, I would like to thank P P Savani University, Gujarat, for hosting this webinar on biosafety and biosecurity. And AIMST University, I think we are very glad to receive 2 students from P P Savani last year, from Mr. Palmer from BSc Biotechnology program, Microbiology program, and Soni Meet Kumar from BSc Biotechnology program. We are very happy to see that well-disciplined, very well-mannered student from P P Savani University. I think congratulations goes to the P P Savani University again. And at AIMST University, we strongly believe that safety of student and it's a prime importance. And while working in the laboratory and beyond laboratory, we must apply sound practices and to protect ourselves and our environment. I think Dr. Subhash has been doing this. He's in charge of biosafety and biosecurity. It is a law by the Malaysian government that we have to adopt these biosecurity features. And Dr. Subhash has been doing this for the last 2 years. This is the 3rd year he's doing it. And the biosafety and security, our university take it very seriously. And we think that it has to be one of the practice we have to do. I think our biosafety and biosecurity month, this time we have a lot of array of programs. I welcome all the students from P P Savani University to join, particularly as mentioned by Dr. Archana, that e-poster session is going to be a very interesting session, and it's a good opportunity for the participant to get gold, silver, and bronze certificates for the e-poster session. So with this, I would like to, I hope you all can come one day to AIMST University and the Global Immersion Program when the COVID pandemic, we overcome that. And with this, I will welcome you all for this particular webinar on biosafety and biosecurity. So hope you all can think safe, work safe, and be safe. Hope this webinar will be enjoyable and useful for everyone. Thank you.
Thank you so much, Dr. Ravi Chandran. Thanks a lot for giving such precious information to our students. We will definitely participate and encourage our students. Now, I would like to invite Dr. Subhash Bhore for his insightful talk. Dr. Subhash, over to you.
Thank you, Dr. Archana. I want to share my screen. So,
Yeah, just let me stop my presentation that way.
Yeah.
All right. Please just go for present now icon.
Yeah. Window.
Yeah. Yeah. And then just in the,
Yeah, you can see, right?
yes, it is starting. Participants, please pin the presentation. That way you can see the slide of Dr. Subhash. So please pin the presentation, okay? You will see the two entries from his name. So pin his presentation, then you will be able to see his presentation.
Okay, it's visible, right?
Yes, it is.
Okay. I just put it on presentation mode.
Yes.
Okay, I'm ready. Okay, thank you very much, Dr. Archana.
Oh, thank you.
Very good morning to all the participants of this webinar. As Dr. Archana mentioned, we are going through a difficult time. I hope your family members, relatives, friends, all are safe and sound. So before I start, I just want to take this opportunity to thank team of P P Savani University for hosting this webinar and becoming a part of our Biosafety and Biosecurity month. So thank you for inviting me to deliver this talk, which is a part of our Biosafety and Biosecurity month webinar series. And before I start about talking, I just want to little bit elaborate about our Biosafety and Biosecurity month. So if you see this slide, because we are organizing this Biosafety and Biosecurity month in association with APSA International, and this one we are doing a second time, and AIMST University is observing October 2020 as a Biosafety and Biosecurity month. The main aim of this exercise is to raise the biosafety and biosecurity awareness among students, staffs, all stakeholders of AIMST University, including P P Savani University students and staff, as well as other participants, those are interested to learn more about biosafety and biosecurity. So altogether, we have organized 10 programs. One webinar is already over, which was on global biocrime and bioterrorism from One Health perspective. So today's webinar, which you are listening to, is webinar number 2, an overview of the Biorisk management from One Health perspective. On October 14, 16, 19, 20, 23, 26, 28, and 30, other programs are there. So I take this opportunity to invite all of you to join this other webinars and poster presentation competition. And also, I want to record my sincere thanks to our AIMST University senior management for supporting this Biosafety and Biosecurity month activities. And special thanks to Dr. Archana for hosting this webinar. I also want to record my thanks to IBC Institutional Biosafety Committee of AIMST University, members for their help and support. last one but not least one, students, those are volunteering to put in place the plan and implement the virus management, biosafety and biosecurity month activities. Okay, ladies and gentlemen, so with this little bit information about our biosafety and biosecurity month, so let's start the talk. So I'm going to give you a very broader picture on overview of the biorisk management from One Health perspective. So since the audience is mixed and the purpose of this biosafety and biosecurity month is to promote biosafety and importance of biosafety and biosecurity, so I kept it very straightforward and simple, but you can always ask questions if you have any at the end of the session. Okay, let's start. So my presentation, I will just give you a little bit background, which will highlight the importance of biorisk management. Biorisk management is extremely important. So I will give you some points to think about. Then I will give you the overview of the biorisk management, and I will touch a little bit to the concept of the One Health, which is very, very important. And I personally believe all the new generation, our young students and youngsters should understand this one and implement and help how they can help in contributing to preserve our environment and contributing in making One Health better. And finally, I will give some take-home messages from the top. Okay, so a little bit background. So if you look at this picture, because there are a number of global challenges global communities facing, that's why the United Nations member states has adopted 17 sustainable development goals to make this world a better place to live. There are a number of challenges, but if you see the topmost three challenges, so these are the topmost three challenges. One is a global warming, because climate change, because of the deforestation, loss of biodiversity, and greed of the humans. So we are losing biodiversity, we are losing forests, and global warming is there because of industrialization and blah, blah, blah. So it is estimated that around 250,000 people may die every year because of the global warming. I'm not going to talk much about global warming and nuclear war, but I just want to tell you a little bit about this background. So nuclear war, many countries are having nuclear weapons, and how many people nuclear war can kill will be decided by the scale of the nuclear war and capacity of the nuclear weapons used. So, but if you see the important component of pandemics or biological risk, so there are many examples, but I'm just giving two examples. For example, the flu pandemic, which was caused by influenza in 100 years ago, that one killed around 20 to 50 million people. Before that, there was a big pandemic. That one is called as Black Death, or it's also called, it was caused by bubonic plague, and that one killed, a rough estimate is somewhere between 75 to 200 million people. So you can understand what the pandemic can do. And there are many outbreaks, examples of outbreaks and pandemics in between. So, and the range of the people killed during these pandemics or outbreaks is somewhere less than 1 million to this example, maximum 200 million. So you can understand what a pandemic can do. So just let's take an example of ongoing pandemic, coronavirus disease 19 pandemic as an example. So this one I just captured day before yesterday. So globally, 35,659,000 plus people are infected with this coronavirus, and more than 1 million people, 1,044,000 plus people lost their life. So if you see the outbreaks, it can be from natural ecosystem, or it can be intentional. Intentional means it can be from the laboratory, or it can be do it yourself, that type of the lab, because nowadays instruments you can buy, you can cultivate the bacteria at home. It's possible. And that's why, I mean, it's possible the outbreak can be natural, or it can be intentional. Natural, it happens because when there is a loss of biodiversity or loss of, deforestation. So as a result, these pathogens escape from that ecosystem, and when they enter into the human population, you have seen example of what's going on now. And intentional, for example, anthrax attacks. So that one is intentional one. So these are some of the examples. So outbreak can be natural or it can be intentional. And doesn't matter it's intentional or natural, the impact is huge. It's terrible. Because if you take the example of COVID-19 pandemic, most of the countries were under lockdown for 3 months or more than 3 months. The global economy is in trouble. And it is estimated that more than 70 million people will be pushed to the poverty because of this COVID-19 pandemic. And it is estimated that we need more than 30 billion US$ over the next year for the development and production of the COVID testing kits or the tests, then vaccine and treatments. So you can understand the huge impact of the pandemic. And that one should help you to understand the importance of the biological risk management. So I'm just narrowing down my talk now. So when you are working in the laboratory, so there are different hazards and there are different types of the safety risks. So I'm going to talk about biological risk, but at the same time, you should be aware of there is chemical safety issues, physical safety, as well as electrical safety. And if you are working in a laboratory as a student or as a researcher or as a scientist or academician, so you should be aware of this holistic picture so you can protect yourself and you can protect others. Okay, so risk in the laboratory environment. So as a student or as a researcher, maybe you are working in agriculture biotechnology, or you are working in healthcare biotechnology, or you are working in industrial biotechnology. So doesn't matter in which sector you are working. So you deal with the genetically modified organisms or non-genetically, non-modified microorganisms, or maybe animal microbes or any type of different categories, and risk is always there. So for this purpose, you should be aware of this risk. And if you take the example of agriculture biotechnology, so it doesn't matter you are doing genetic engineering of plants or animals. So your work starts with the genetic engineering of the microorganisms, because you have to put the promoter, gene of your interest, terminator in order, and you have to make the expression cassette to make your vector ready for the transformation. So I think in India, I'm not sure Blue Rose is there or not, but you can see the power of genetic engineering. So this one is developed by the Australian scientist by using the inverted repeats, intron spliced inverted repeats, and changing the pathway. So instead of having regular colors, we can have the blue color rose, which is not available in the market. And maybe you are working on domesticated animals, genetic engineering, because huge number of people are enjoying meat of goat or sheep or whatever it is. So when you are working on genetic engineering to increase the meat mass or to increase the production, so you have to deal with these genetically modified microorganisms, and risk is always there. Or in case if you are working in diagnostic labs, or maybe you are working in somewhere in the healthcare sector. So if you look at this list of the viruses, so these 15 viruses, so they have killed a huge number of people in the past, and especially those are highlighted in different color. So they have done a huge economic loss, social impact, as well as other losses. And now this new virus, new strain of this coronavirus is there, and that one is making life difficult in every country, and that's why we are having this webinar instead of having face-to-face. So in addition to that one, outbreaks can be from bacterial pathogens. You take the example of cholera or other examples, or it can be a fungus. For example, this multi-drug resistant Candida aureus. So yeah, it doesn't matter you are working in agriculture biotechnology or healthcare biotechnology or industrial biotechnology, there is no sharp dividing line between these sectors. So you directly, indirectly come in contact with all this type of work when you are working as a scientist or researcher or as a student, and you should be aware of all these risks. So in healthcare, because if you know human diseases, altogether, I mean, 60% of the human diseases are zoonotic. What does it mean? Means all these originated from the animals. So when the pathogen escapes from the animal population to the human population, so as a result of that one, we will have the diseases, and that one is a 60% are zoonotic diseases. And you will be surprised to see these figures or facts. Around 2.4 billion cases are registered every year because of zoonotic diseases, and around 2.2 million deaths per year. So it highlights the importance of also biological risk management, because all these diseases are caused by either viruses or bacteria or parasites or the fungus. And as far as the laboratory safety is concerned, because when you are working in the laboratory, maybe you are using test animals or testing some drugs, either designed, excuse me, either designed by using molecular modeling, or maybe you have isolated something from medicinal plants or from the deep forest and you are testing animals. So, I mean, in addition to that one, the disposal of the waste, maybe you are testing some bioactive compounds, efficacy or the effect on some pathogens, and when you are not disposing the waste properly, there is always chance it can escape, or if it's not escape, if you are not taking proper care, you can get infected, and that one is called as lab acquired infection. So in addition to that one, unsterilized items, microbial cultures which you have in your cold room or deep freezer or in the lab, and these risks are always there. So if you see the whole spectrum, it's a very broader picture. So in this case, because of deforestation, because of the loss of biodiversity, and all other human activities, new emerging diseases are popping up. And in addition to this chronic, diseases and emerging diseases, we have to make sure that these new pathogens or these deadly pathogens should not fall in wrong hands. And that's why biorisk management also helps to make sure that there will be no dual use of the technology, because we have to make sure that these chronic diseases or the emerging new diseases will not lead to the disaster, and we can make sure that there will be no likelihood your right hand side means bioterrorism or biowarfare. Then in addition to that one, there are benefits. So all this is important and needed, and especially the youngsters, new generation like you. Most of the participants are students, so you should be aware of this one to protect yourself as well as others, and keep this planet safe for the next generation. So this one is just a holistic picture to tell you that how it is important, because this one is a very broad picture. So if you see at the bottom, so this one is United Nations. So altogether means 193. Okay, I think currently 194 member states are there, and when they implement this different international guidelines or laws or different policies, so then individual government implements these policies, and then it comes to the university or the center of excellence or research centers, and as implementation body or committee, institutional biosafety committee, implements all this what is needed to keep the students, staff, stakeholders, and all safe from the biological agents. So if there is nothing, if everything is fine at institutional level, so there should be no reason why there should be an outbreak. So that's why implementing the policies, putting everything in place for make sure that there will be no lab acquired infection, or there will be no misuse of the pathogens or biological agents from the laboratory. The role of the institutional biosafety committee, as well as those are working in the laboratory, is very, very important, especially the principal investigators. They should know what's going on, and their students are safe, and that one is very, very important. So if there is no problem at institutional level, so that means we should not have a problem at national level. And if all the countries are implementing the right guidelines, policies, and they have acts in place, so as a result of that one, there will be no problem at national level. And if all the countries are serious about the biosafety and biosecurity or biorisk management from One Health perspective, then there should be no problem at the international level. That means we are safe. So as a result of that one, we will have a global biosafety and biosecurity, and it is very, very essential because now you are experiencing, all of us are experiencing what is the impact of the pandemic, and we are everybody is suffering. So just to highlight, because at global level, how we protect the human population from these pandemics and different types of diseases. So there are many different guidelines, policies, treaties, conventions, and international acts are there. So for example, if you take Biological Weapons Convention, then Convention on Biological Diversity, because biodiversity is important to protect the biodiversity. Then we have Cartagena Protocol on Biosafety, which deals with the movement of genetically modified organisms from one country to another country, or transboundary movement. And the next one, the International Health Regulation. This one is very, very interesting because the WHO coordinates all these activities, and international health regulation is implemented to prevent the spread of the diseases from outbreak site or the country where outbreak happened. So it should not spread to other countries. But I think the whole world now realize there are loopholes or the gaps in the international health regulation, and I think nobody will deny. So I think once the pandemic is over, the people, those are involved in drafting the international regulations, they will work together and they will fix the loopholes to make sure that this type of the scenario will not, this type of the pandemic situation will not happen in the future. So in addition to that one, we have a WHO Biorisk Management Framework, and as well as we have a United Nations model regulation, because dangerous goods regulation. So for example, shipping the samples or transferring dangerous pathogens or the biological agents from one country to another country, or from one research center to another research center, or one collaborator's laboratory to another collaborator's laboratory. So all these different policies, guidelines, treaties, conventions are in place to protect all of us and the environment, as well as to keep everybody safe. So if you think about the risks involved in biological laboratory, so biological hazards pose risk not only to the researchers, but in case if it by mistake escapes from the laboratory, so it is dangerous for the surrounding community, because towns are around the university as well as the center of excellence, and if they get infected, so in addition to that one, the environment, agriculture, crops, domesticated animals. So it's a very complex chain. So for this purpose, we should always, we have to practice the biorisk management to keep or contain the microbes and pathogens of the humans, animals, as well as the plants. Okay, so risk versus biological risk. So what is meant by risk? risk is because risk is everywhere in our daily life. Because, for example, when you early in the morning get up, once you take bath, and once you go out, so when you are crossing the road, there is always chance car can hit you, and there can be accident. So risk is always there. So how we can define the risk is it's nothing but possibility that something bad or unpleasant will happen. And the overall risk is calculated by the likelihood and the consequences. So for example, likelihood, you go in the zoo, so that the tiger or lion will not attack you. But if you go in the forest, and if tiger or lion see you, so likelihood is very high, and the consequences are deadly. You may not come back from the forest. But if you are going in zoo, so the likelihood is low, and the consequences are very less, because you are going in zoo, and tigers and lions are kept in such a way that they will not attack you. So risk depends on the situation. This one is very, very important. Depends on the situation. That's why I gave example of, if you are going in forest, and if you see the lion or tiger, and if you are going in zoo, and the likelihood is different, consequences are different. So it depends on the situation. Okay, so in addition to getting exposed to the infectious agents or biological agents while working in the laboratory, and when the pathogens escape and goes into the surrounding community or the environment, but in addition to that one, there is also a risk of somebody can steal the pathogens from the laboratory. They can misuse it, and maybe they are having a link with the terrorists. So cumulatively, not only the exposure of the researchers, but stealing, misuse, and other risks are always there. So all these risks involving biological agents are collectively called as biorisks. And how to deal with this biorisks, I will tell you in short time. So broadly speaking, I just want to make sure that you are very clear about the fundamental difference between the biosafety and biosecurity. So what is the fundamental difference between biosafety and biosecurity? Biosafety is nothing but protecting the researchers, those are working in the laboratory, from the pathogens or the microbes, so that they will not get infected. So that one is all about the biosafety. So you are using different types of the control measures to make sure that they will not get infected. But biosecurity is opposite, means you are protecting the microbes or pathogens from the bad people. So they will not get hold of the pathogens, and they will not misuse it, or they will not pass it to the wrong people for the different intention. So when you are protecting the microbes from loss, theft, misuse, and intentional unauthorized access or release, so that one is called as biosecurity. So this one is a fundamental difference between the biosafety and biosecurity, because generally the researchers think that biosafety and biosecurity is same. Some common elements are there, but they are not identical. So that one you have to keep in mind. Okay, so in case of the biosafety, so it happens because of the accidental exposure. And in case of the biosecurity, it happens because of the misuse or theft or diversion, or in some cases sabotage. Maybe somebody unhappy individual is working in the laboratory, and that person want to take the revenge. It's possible. There are many examples in the history or in different other countries. So that one can happen, and that one is a part of the biosecurity risk. That one is called as insider threat, insider, outsider. Maybe somebody from outside can come in the lab and can steal, or maybe somebody who is working in the laboratory can steal and can misuse it. So insider threat, outsider threat. So these are the fundamental elements of the biosafety risk and biosecurity risk. So now the important question is how we can deal with biosafety and biosecurity risks. So the answer is effective biorisk management. That's why the biorisk management is very, very important. Not only important, it's essential to make sure that all of us are safe. And I think you will be convinced because all of us are experiencing the effect of the coronavirus disease pandemic. So that's why the effective biorisk management is very, very important. So what is biorisk management? So effective, biorisk management is nothing but the effective management of the risk which is there in the laboratory while working with the infectious agents, toxins, or their derivatives. And it involves number of practices and procedures to contain the biological agents or to ensure the biosafety and biosecurity as well as the containment. So this one is a beautiful definition. So this the reference which I have given at the bottom. So this one biorisk management is based on this document. So this one is developed by the international community and it's well accepted by the global community. So what we need for the biorisk management, another important question. So biorisk management, everybody knows it's very important, it's essential, and each and every university or center of excellence or company, biotech company which is involved in modern biotechnology activities should implement the stringent procedures for biorisk management. So what we need for the biorisk management? We need a full support from the top or senior management of that institution, maybe it's a university or any type of research organization. We need a full support of the senior management. Otherwise, it's impossible to implement the biorisk management. At the same time, we should have a very competent team of institutional biosafety committee because they are considered as expert in that area and guiding the principal investigators, guiding the students, as well as putting the things in place to make sure that nothing will go wrong in the campus. And at the same time, competent biosafety officers we should have. So in case if we don't have competent biosafety officer, then it will be very challenging. Then biorisk management core documents, for example, biorisk management manual for the institution or for the university. We have developed for the AIMST University, but we are still in midst of implementing biorisk management since we started last year. So for example, biorisk management manual, different types of SOPs, how to handle the autoclave, how to dispose the waste material, as well as SOPs for the other procedures in the laboratories. We generate the aerosol. So these different types of the documents should be in place. Then safety policy of the university or the institution policy should be in place, planning, implementation, and operation. So that's why the roles and responsibilities are very, very important. Biorisk management cannot be implemented by one person. It's not a one person's job. Everybody should play their role. Everybody should take the responsibility. Everybody should take the accountability. And for this purpose, the roles and responsibilities are very, very important. And everybody should be on the same page. So because, for example, what's going on in the university or institution, students should be made aware of that one. They should not be in dark. Principal investigators should know, that's why I'm saying everybody should be on the same page. Then checking and corrective actions. And finally, the biorisk management review to find out the gaps in the biorisk management and fixing the gaps to make the system better, efficient, and ensuring the safety and security of everybody. Okay, so biorisk management, if you see the broader picture, so biorisk management can be divided into three primary components. Number one, assessment. Number two, mitigation. And number three is performance. So these are the three fundamental pillars of the biorisk management. And if you see this picture, so it's just like a tripod. I think everybody, either in the lab or at home, you are using this tripod. So if the one leg is broken of this tripod, so this tripod will collapse. So similarly, so this assessment, mitigation, performance, so all these three pillars are equally important. Not only assessment is important or mitigation is important. All three are equally important. So this biorisk management, AMP model, which is developed by the WHO, So this one basically is based on the plan, do, check, act cycle. On your right hand side, what you see, plan, do, check, act. So this one is also called as Deming cycle or Deming circle or Deming wheel. So the whole biorisk management is based on this concept of the AMP model and plan, do, check, act cycle. So it's not a linear, it's a circular, means AMP, assessment, mitigation, performance, assessment, mitigation, performance, assessment, mitigation, performance. So the cycle goes on till you make it perfect, and there is no problem in the campus. So same goes with this plan, do, check, act, plan, do, check, act, plan, do, check, act. So until and unless you solve the problem, you can just continue, and it's not a one-time exercise. You have to do regularly. It depends on the situation. So these are the common steps how you can do the biorisk management. I mean the steps in all in this process. So first of all, for the understanding, it can be a project specific, or it can be a institution specific, or it can be a facility specific. So first of all, you have to define the situation by asking fundamental questions. For example, what is the hazard in the laboratory? Or how lab procedures are completed? Is there any chance of generating aerosols? And who is the host? Does it infect the animals? Does that pathogen infect the humans? Does it infect the other host? So you should know the range of the host. In case of the, you have to do the, risk assessment, or the you have to see through the biosecurity angle also. For example, who is the threat? Some time ago I have mentioned it can be outsider, it can be insider. Who is the adversary in that situation? Outsider, insider. If it's insider, who? And you have to fix the problem in time before it becomes late. Then how? Do we have a CCTVs in place? Do we have a security guard and security system in place? Are your, do you have your assets, means pathogens or glycerol stocks or whatever it is, well protected? So that one, you need to ask these fundamental questions, and you have to characterize the risk. And after that one, you have to determine risks are acceptable or not. If the risk is not acceptable, you have to do something. You have to put the additional control measures. You have to take some actions to minimize the risk, and then you can proceed. Otherwise, you are bound to have some disaster in the near future or in the future. So for this purpose, this one is very, very important. Okay, so when you are working in the laboratory, so when you are doing the risk assessment, because I told you these fundamental three pillars: assessment, mitigation, performance. So in that case, when you are doing the risk assessment, so you have to do the risk assessment separately for the biosafety, and you have to do the risk assessment separately for the biosecurity. So when you are doing the biosafety risk assessment, you can use this 5 P's approach. So this 5 P's stands for pathogen. You need to think about the pathogen, procedures, place, people, those are working in the laboratory, as well as personal protective equipment, which types of personal protective equipment you need, then their maintenance status, if you're thinking about the pathogens, infectious dose, as well as routes of transmission. Routes of transmission, those of you are not clear how, if the pathogen is not entering in your body, there is no reason why you should be sick. So when the pathogen enters in your body, so as a result of that one, you fall sick. So there are 4 ways how pathogen can enter inside your body. One is inhalation of the contaminated air, which is with the pathogen, something like this. So when you take the contaminated air, so you get infected and you will have the disease. Another one, contaminated food or drink. So that's why many people, those are eating partially cooked meat or not properly cooked meat. So generally they will have some infection. So contaminated food, or maybe it's a contaminated water. Then third one, mucus. For example, if you are working on HIV and a very small, tiny blood drop just escaped while doing pipetting or while doing some procedures and just landed on the mucus in your eyes. So as a result of that one, you will have the contraction of the HIV. I mean, of course, if you take the action in instantly or quickly, there are chances you can recover. But entry point I'm highlighting. So mucus. Then number 4 is means maybe you are having some cuts or wounds on the skin, and when the pathogen comes in contact with that wound or that part, so the pathogen can enter. Just like when there is a no guard in front of the main gate of your university or your research center or your maybe school. So in that case, anybody can enter. So it's just I mean, same principle. When there is a cut in the skin, so pathogens can enter easily. Okay, so this one is for the biosafety risk assessment, and this one is a group exercise, but this one is a webinar, so it will be difficult. But maybe later on, I'm going to give all the slides to you, so don't worry. This one you can just go to this website and you can download this. I think you can see. So it looks like this one. So once you open this one, you will see screen like this, and you can get lots of information from this risk group database, which is made available by APSA. So this one, the group exercise, maybe you can do it, because generally I ask students to do this one, enter the E. coli, Bacillus anthracis, Ebola virus, and I ask different questions. So this one, maybe you can do it later. So this one definitely will help you to learn more about the risk groups and additional information. Okay, so you also need to do the biosecurity risk assessment. Biosecurity risk assessment is very, very important to make sure that there is no loss, theft, or misuse of the pathogens. Those are deadly, as well as there is no loss, theft, and misuse of the biological material, or maybe very sensitive information which is related to the ongoing research project, or maybe some protocols to make some toxins or whatever it is. So that one, all these things can be done by implementing the policies, procedures, as well as tracking inventory. Tracking inventory means, for example, you have a glycerol stock in deep freezer, you have in append-up tube, which is 1.5 ml. So if somebody is taking 5 microliter or 2 microliter, that one is good enough to go back and generate a huge amount of the cultures or huge amount of the pathogens. So for that purpose, tracking the inventory is important. Who is having access to the deep freezer or these pathogens, that one is important. And that one is part of the biosecurity. That's why tracking the inventory, then limiting and monitoring the access. Not everybody should be able to open the deep freezer and see what is there and what type of pathogens are there. So the access should be limited. Not everybody should be allowed to open to have access to these pathogens. So that's why the everybody should be on the same page, and everybody should work for the biosafety and biosecurity of that laboratory or institution or the facility. So this biosecurity risk assessment you can do by this simple 5 steps method: identify, assess, analyze, design, and develop, and evaluate. So with the help of this one, you can find out where is the gap in biosecurity of that university or institution, and what type of control measures you can put in place to fix the loophole to make sure that disaster will not happen. Then your biosafety and biosecurity risk assessment, when you are doing, you should do a 360-degree analysis. So this one example purposely I'm giving because if you see this news, it happened in New Delhi, India. So this one happened in last May. It's not just few months ago. So the monkey escaped with the COVID-19 samples after attacking the lab assistant. And later on, that monkey was spotted chewing the sample kits. And this one is a good example. So not only biosafety, but biosecurity is also very, very important. So can you imagine something like this one happens, and when you have the cultures of the dangerous pathogens. This happen in Delhi with the coronavirus samples from the COVID-19 patients, and that's why when you are thinking about the biosafety and biosecurity of the facility or laboratory, you should think you should do the 360-degree analysis. Okay, let's move on. So when you find out or when you do the robust risk assessment, so you will know what is the gap in the system, and based on that one, you can put in place the right control measures. So there are 5 different approaches which you can use to mitigate the risk. So first one is elimination, second one is substitution, third one is engineering controls, fourth one is administrative controls, and fifth one is personal protective equipment. So if you see the hierarchy, means which one is effective, which one is less effective. So if you see this triangle, which is very popular triangle hierarchy. So personal protective equipment, for example, you take the example of the mask, which you are using to protect yourself from the coronavirus. So that one is one example of the mask which you use. And elimination, for example, if you don't have the facility in the laboratory to handle the coronavirus, or if you don't have the facility to handle, for example, Ebola. So you should not bring Ebola sample in the campus. Elimination means make sure that that one is not there physically. Elimination. Substitution. So for example, those of you are working on CDNA library construction, so it's not necessary you should use the pathogenic culture to make a CDNA library. Just use the simple one, E. coli. So you are using E. coli for the CDNA library construction. Substitution. But at the same time, you can use other dangerous bacterial pathogens for the CDNA library construction. So this one is an example of substitution. Engineering control, you are using the biosafety cabinet and other different types of the engineering controls to protect the researchers. Administrative control, maybe I will give some examples. For example, everywhere when you go now, they check your temperature. If the temperature is within 36 to 37, you are consider okay, it's normal temperature. But if it's going beyond 37.3 or above, then we can find out some risk is there. Another one, that's why in addition to temperature, physical distancing. We are asking and we are recommending to everybody there should be at least 1 meter distance. But in crowded countries like in India, it is very difficult to practice and it's very challenging. So, but physical distancing, then a temperature check, then a regular hand washing. So all this, these are administrative controls. So this one is one example, and PPE, the mask which you use. So these are, some control measures. So if effectiveness, if you see, so the PPE is less effective. Elimination is the most effective, because if you're not bringing the dangerous pathogen in campus, there is no need to worry. Or maybe I will give another example. So shark is dangerous. Shark is dangerous, but if you're not going in a sea where the sharks are there, so then you don't need to worry about the risk posed by the sharks. So that's why elimination is the most effective. And in case of the PPE, it's the least effective, but it's very, very essential. Okay, so let's move on. So then this biorisk management performance. This one is the third pillar of the AMP model: assessment, mitigation, performance. Assessment, mitigation, performance. So this performance, this one is the third fundamental pillar of the AMP model. So this one is just like grading the student. So for example, academicians, those are in this virtual room and listening to me. So we do marking of the answer scripts, and we find out how many students are in A, how many are in B, how many are in C and D. The system may be different. This one is based on the American system. So in system in India, maybe a little bit different, or in Malaysia, maybe it's a little bit different. So in this case, what we are doing, how the biorisk management system is performing, whether it's outstanding, or it's unsatisfactory, or it's very good, or it's just satisfactory. So that grading can be done by doing performance evaluation, and that one is very, very important. That's why I told you this assessment, mitigation, performance. all these fundamental 3 pillars are equally important, and that's why you have to do the performance sincerely to find out how it is working. So this performance is a third pillar of the biorisk management model, and it helps you to minimize the risk when you are, for example, there are many lab acquired infections. So that means something is wrong in the laboratory, or maybe there are some accidents happening again and again in the laboratory. That means something is wrong in the laboratory. So you have to see the performance based on number of indicators, and based on that one, you have to implement the mitigation measures to make sure that it will not happen, or you can minimize the risk, the level of the risk, to make sure that it's within acceptable range. So this, I mean, there are a number of tools how you can do the biorisk management performance. You can do the audits. Audits can be done annually or 6 monthly or quarterly, depends on situation. Because if the facility is A4, BSL4, then risk is very high. Or you are starting new project, so then audit can be done. I mean, it's not necessarily you should wait for 1 year. So you can do maybe once in a 6 month. Then inspections you can do regularly in a standard format, or maybe it's a spot check. Without informing anybody from the laboratory, you can just go and see the real situation. But in that case, there will be a shock for the students and the researchers, those are working there. So each and every this tool do have some advantages and disadvantages. So for example, you can take the, you can interview the researchers or students, those are working in the laboratory, and you can find out the gaps. And training is done, workshops are conducted, then you can see the efficacy of the workshops. And incident report, this one is important, number 6, incident reports. So when there is accident, something broken, or maybe you have by mistake, conical flask or the universal bottle or whatever, in which you are culturing the microbes or the bacteria, slipped from your hand and it's broken. So all the microbes are everywhere on the floor. So in that case, incident reporting is very important to find out the root cause. But in many laboratories, I'm not talking about Malaysia, I'm talking about the overall situation. So incident report, students or those are involved in incident, they are reluctant to report it, because they think laboratory manager will penalize, or supervisor will scold and penalize, or management will be very angry. So they don't report the incident. But that one, incident reporting should be encouraged. So as a result of that one, we can identify the gaps and we can make the system better for this purpose. So these are the different tools. So just little bit I want to touch upon on One Health. So this One Health, because we need best life, that's why we are developing, we are doing lots of things. As a human, we are different as compared to animals and other organisms. So but only human health, if we are thinking, it's not right, because humans cannot be healthy if the animals are not healthy. Humans cannot be healthy if the environment is not healthy. Animals will not be healthy if the environment is not healthy. So it's a complex thing. It's a complex situation. So for this purpose, in order to have a better health, in order to enjoy the better health, so environment should be healthy, animals should be healthy, and for this purpose, global community need to think about this one, and this whole concept where there is optimal health for humans, optimal health for environment, and optimal health for the animals. So this optimal health for these 3 components, it's called as One Health. And global community need to, global community is promoting this One Health concept to make sure that we'll not have pandemics like this, will not have outbreaks time to time, as I mentioned before. So biodiversity conservation, minimizing the deforestation, and minimizing the impact of the climate change. Maybe small islands or small countries will go under the water when the temperature is increasing. So this One Health concept we need to promote, and it's a very, very important for the global community. And on top of that, this effective biorisk management can serve as a gear to boost the One Health. And as a result of that one, if the One Health is wonderful, that means human population is healthy, animal population is healthy, environment is healthy. So as a result of that one, it will boost the global sustainable development, and it is very, very essential. And in line with the global agenda, 17 sustainable development goals, those are accepted by the global community. So to achieve this 17 sustainable development goals, so the One Health is very, very important, or it's a key component for the sustainable development. So by the 2030, we were supposed to make this world as a better place by implementing the global agenda and to achieve the goals under 17 sustainable development goals. But now, because of this pandemic, outcome could be different because the global economy is in trouble. So, but I want to make sure that all the participants, those are in a virtual room, understand the importance of One Health, and you can share with others when you go back. So finally, I just want to give the take-home messages. So the message number one is biorisk management is entirely based on PDCA cycle, means plan, do, check, act, plan, do, check, act, and AMP model, AMP, assessment, mitigation, performance, assessment, mitigation, performance. Number two message is the ultimate goal or the main aim of the biorisk management is to protect humans, animals, and environment. Number three message, the number third message, take-home message is implementation of biorisk management is essential for the local, national, regional, and global safety as well as security. And last one but not least one, we need to promote, all of us, we need to promote One Health concept, because as a human, I mean, we cannot be healthy if the animals are not healthy and environment is not healthy. So with this message, I stop my presentation, and I want to thank everybody. Thank you very much for your attention, and I will be more than happy to take some questions if you have.
Okay, thank you so much, Dr. Subhash. Participants, do you have any questions? Please feel free to ask if you have any questions. Okay, so I would like to extend my thank you to Dr. Subhash Bhore for very informative session today. I'm sure your talk is, quite relevant in current time, and especially when entire world is struggling against COVID outbreak. This session was specifically very insightful for all of us, and I'm sure my students have learned, and all the participants have learned a lot about biorisk management. Now, I would like to invite our principal, Dr. Anish Sharma, sir, for his vote of thanks for the event. Sir, please. Can you hear me, Dr. Anish?
Thank you, Dr. Archana. Hello. Thank you, Dr. Archana. On the behalf of P P Savani University, I, Dr. Anish Sharma, Principal, School of Sciences, expresses my sincere thanks to Dr. Subhash Bhore for conducting such an informative webinar on biorisk management and biosafety aspects. This was really a very informative webinar, and it has enhanced not only the knowledge of our students, but also, I think, of the academicians and researchers who are the participants in this webinar. Thank you, Dr. Subhash, and I am hopeful that we will conduct many more webinars uh, in collaboration with AIMST University, Malaysia. And thank you, sir. Thank you for the conduction of such an informative session. And I hope whenever I also invite you to visit P P Savani University whenever you visit India. Thank you, Dr. Bhore.
Okay, thank you, sir.
Thank you, sir.
There is a question from one of the participant, our student, Vivek. He is asking, I will read it for you, Dr. Subhash. Is it okay?
Yeah, please go ahead.
Okay. How can we eliminate the things? Because we as biotechnologists need to help overcome the problems of society like coronaviruses. Can you please highlight more on elimination, like how to eliminate the problem altogether? Is there is a way? That is what I think he wants to ask.
Okay. Thank you, Dr. Archana. Thank you. I think it's a student from your university. So it's
Yes.
a wonderful question, because that one is one of the mitigation measure. Elimination, maybe I will give one example so it will help that student to understand or others to understand. Elimination, for example, in a traditional way, we have a biosafety level 1, biosafety level 2, biosafety level 3, and biosafety level 4. So the deadly pathogens for which we don't have the vaccine, those are deadly, like those don't have vaccine or those who don't have any treatment. So that one we always handle in BSL 4, because there the control measures, the filtered air, as well as security measures, as well as biosafety measures are very, very stringent. So, but if that type of pathogen you are bringing to BSL 1, so definitely people, those are working there, will get infected. So for that purpose, elimination means you don't bring that type of pathogen in BSL 1 or BSL 2 because you don't have facilities. So that work needs to be carried out in a facility where you have all the control measures in place, like BSL 4. So that's why that one is, hope this one will help you to understand. Elimination means you are not bringing that pathogen or dangerous biological agent in the BSL1 facility where you don't have enough facilities to deal with that one or to contain that one. So that one is one example I gave you to explain. Elimination means you are not bringing that one in campus. So I gave example while talking. If you are not going in a sea where the sharks are there, so there is no risk. So similarly, when you are not bringing pathogens in a BSL1 facility or facility where you don't have the control measures in place, so that's all about elimination. I hope I have answered your question.
Okay, yeah, thank you so much, doctor. Okay, yeah, I think Vivek said thank you, sir. And there is one more query from one of my student, Dhruvi Damelia. She is asking what happened to animal after the first vaccination trial. She wants to know.
Oh, very interesting question. So, because when we are developing the vaccines, we don't take directly to the market and make it available through the pharmacies or through the hospitals, because we have to do the testing on the animal models. And in the process, those are animal lovers. Always they don't like it because animals will be get killed in the process. And but we don't have any option, because before using these vaccines or drugs for the humans, we need to do the animal testing by using the animal models. Maybe it's a chimpanzee, or maybe it's a rat or a rabbit or horses. It depends on the objectives of that exercise. So in that case, when the animals are, those are, killed during the process, those are, I think killed word is not right, euthanated. So those are sacrificed or euthanated. I think that one will be right term. So in that case, the disposal of the dead dead animal bodies, it's very, very essential. Because, for example, if you are putting the animal organs or the animal body parts just in a simple way, without proper disposal procedures or without using a right SOP. So in that case, because flies can sit on the meat of the animal, as well as it can disseminate through the insect. So the disposal, these animals, those are used in vaccine trials, are disposed very, very carefully with the extra care to make sure that that pathogen will not escape environment, or it will not escape into the ecosystem. So that proper disposal is very, very important when that type of trials are going on, because we never know how it will escape. I hope I have answered your question.
Yeah, Dhruvi.
Hmm.
Okay, I guess. Anybody else has any more queries? Dr. Subhash is an expert. Feel free to ask. You will get to know a lot of things. Okay. Okay, participants. So make sure to fill the feedback form, which I have shared with you, okay, during this session. And I'm also going to share after this webinar, I'm going to share, the information about all other sessions organized and hosted by AIMST University, and also the online e-poster competition. I have told you in the beginning about that. So make sure to participate into that, because for sure you will learn a lot, you'll get a good exposure, and you'll get rewarded as there are grades for it. So say for example, if 10 students are falling in category A, grade A, all those A's will get gold medal, then silver, and then bronze. So it is a good opportunity. All right, there is one more question from me, Sony, one of our student. Uh, so he's asking, well, we had done biosafety with hands-on session. Okay, that was great. Okay, he is just sharing his experience. He is not asking anything, but he went. He and Heth Parmar, both of my students, they went last year. I mean, this year, in the beginning of this year, for short-term global immersion program, and they have learned a lot of hands-on uh, biosafety sessions. So he's saying that it was a great experience to learn at your facility, Dr. Subhash.
Yeah, thank you very much, Dr. Archana. I'm happy to hear that because during that time we conducted some sessions in the laboratory just to give them feel, but that one was just tip of the iceberg, Yeah.
Yeah. So shall we wrap up now
Yeah,
here?
yeah.
Okay, thank you so much once again. Thank you, participants. All right. Okay.
Dr. Archana,
Yes.
did you show them link or QR code so they can download the slides immediately and they can fill in the feedback form?
Yeah, I have shared the feedback link already to them. If you want, I can also share the QR code. If you have that slide with you, the QR code, please display on your screen.
Yeah,
Or,
yeah,
yeah,
yeah, I can. Yeah.
yes.
Just a few seconds.
Yeah. So, participants, there is a quick way of getting, your attendee certificate. Dr. Subhash Bhore is going to display a barcode. If you have a scanner, you can scan it, and right away you will get a link. That's going to be a feedback link, and once you fill it, you will get the certificate. Okay, so make sure to do that. And if you have any other information, you can always,
Yeah, this is
contact us. Okay.
Dr. Archana. You
Yeah.
can
So
see.
if you
Can you
have
see?
a QR scanner, please do that. That way, it is even more easier. You know,
Yeah.
technology rocks. This is the era of technology. So
Yeah, I request all the participants to fill in the feedback because we are just measuring the impact of the webinar. So I will request all the participants just to fill in this feedback form. if you want to download the slides, you can just scan this one and you can download, because I want to make sure that once you download, you share with your, other friends or everybody. Just share with everybody. Everybody should know about biosafety and biosecurity.
Yeah, so as I said in the beginning of our joint session, that it is going to be, this webinar is mainly hosted to spread the awareness about biosafety, biosecurity, and how to take care of those things. So make sure our society is very well aware of these things. So that is one of the major issues these days. So please spread the words among your friends. All right. Okay. That's all for today. Okay, students. Okay. Thank you so much, Dr. Subhash. Once again, we really want to thank you for a splendid session. Thanks a lot. Okay, everyone. Okay. Bye bye. Have a great day. Okay. Bye.
Bye, everybody. Thank you.